Hello, my name is Kyle, and in this presentation, I will be talking about the enzymology and anatomy of fat absorption. Now, the figure to my right represents the human digestive system, and all macromolecules, whether that is carbohydrates, protein, or lipids, pass through this system. Through the process of chewing and swallowing, chewed up food, otherwise known as bolus, will will flow through the esophagus. The esophageal sphincter will open, allowing bolus to enter the stomach. Bolus further gets broken down into watery chyme, and the chyme then enters the small intestine via um, the pyloric valve. So the pyloric valve will open, allowing small amounts of chyme to enter the duodenum of the small intestine. And it is in the duodenum of the small intestine where most nutrient absorption takes place. Now, in general, when the food that doesn't get, or the nutrients that don't get absorbed will continue to flow through this tubular structure um, from the small intestine into the large intestine, and it's here where the chyme turns into semi-solid feces. The feces will travel down the rectum and out of the anus to be excreted as waste. Now, on the left-hand side is a flow chart that I created to illustrate fat breakdown, particularly the fat breakdown that we will be talking more specifically in this presentation. So the ingested food from fat globules will travel down into and group together as fat droplets. Now we're in the small intestine. Through enzymatic activity and other key components, the fat droplets will turn to my cells. The fat components that make up my cells, they will enter the intestinal cell as monoglycerides, free fatty acids, and then recombine into triglycerides. Then the triglycerides will group together with cholesterol and protein to form lipoproteins known as chylomicrons. These chylomicrons will then exit the intestinal cell and get absorbed into the lymphatic system. We will go more in depth with this mechanism later in this presentation. But for now, I wanted to touch briefly on the overview of lipid digestion and absorption. So, in a typical human diet, 90% of the fats that we ingest, triglycerides are its primary form. Lipids are different based on the makeup, the natural makeup of lipids. They are different from carbohydrates and protein in that they are not water soluble. And this poses a significant problem in digestion and absorption in that enzymatic activity, they have a difficult time breaking down lipids because lipids do not mesh well with the our body's aqueous environment. Instead, lipids will group together to form large fat droplets. So, what do we do about it? This is where lipase comes in, and lipases are enzymes that break down fat into smaller substrates, smaller components. Now, fat breakdown or digestion really begins in the salivary glands. So the salivary glands will secrete lingual lipase for fat digestion. Then stomach, as food continues to flow down the esophagus and into the stomach, gastric lipase gets secreted from the stomach. But really the pancreatic lipase, which I didn't box here, but the pancreatic lipase um, uh, is where most of the fat breakdown can occur. And the triglycerides get broken down into a monoglyceride and two free fatty acids. Okay, in this slide, I really wanted to focus on the key accessory glandular organs that play a huge role in fat absorption. All right, so the first one that first organ we want to talk about is the liver. So the liver produces and secretes bile, and one of the components of bile is bile salts. Now, bile salts play a huge role in fat digestion because they are amphipathic, meaning that they have hydrophobic and hydrophilic regions. Bile salts then can attach to the fat droplets and coat the lipid molecules, creating a stable emulsion of smaller fat droplets. So if you can see in this figure here, the bile salt is binding with a lipid droplet, and the polar regions are of the bile salts are combined, I guess, or closer to like the aqueous environment here, here on the outside. The next organ that I wanted to talk about was the pancreas. Now, the pancreas secretes lipase and colipase. As I mentioned earlier, pancreatic lipase breaks down fat into smaller substrates, but colipase is a coprotein that enhances lipase activity. And by that, I mean that it anchors the lipase, which allows the colipase will remove some or will displace some bile salts, which allow the lipase to continue to break down fat and really dissolve, go into the center, I guess, of the fat molecule to break it down into further smaller fat substrates. Now, the glandular organ that I didn't mention in this slide is the gallbladder, which is this green structure that you see right here. The gallbladder's role is to store excess bile, and when the human consumes fats or fatty foods, this the bile that gets stored in the gallbladder can secrete out, travel down the bile duct and into the duodenum, which will allow for more bile salts for further fat digestion. Okay, now that I've given you the background information of all of the necessary structures, it's time to really go and dig deeper into fat absorption. So this slide showcases fat absorption from the small intestine to the enterocytes. And what we see here is that the bile salts will attach onto the surface of the large fat droplets from the stomach, and then turn the fat droplets into smaller, will emulsify them into smaller particles. The bile salts, I forgot to mention, act as detergent. From there, in number 2, pancreatic lipase and colipase will break down fat into monoglycerides and other free fatty acids. And these monoglycerides, free fatty acids, because of their hydrophobic nature, they will group together into micelles. And micelles are just aggregation of fatty acids, mono and diglycerides, phospholipids, and cholesterol, Okay, and then from there, these individual, I guess, fatty acids and monoglycerides from the micelles will enter the interiocyte through the apical side of the intestinal cell. So in the case of 3A, um, the the monoglycerides and free fatty acids will pass through simple diffusion. Cholesterol is a little different. Cholesterol will pass through via the cell through a transport protein. Okay, from here, the interiocyte to the lymphatic system. In number 4, once the cholesterol and the free fatty acids enter the intestinal cell, well, the smooth ER will recombine these monoglycerides and free fatty acids into its natural form, the triglycerides. Then cholesterol, triglycerides, and protein will form chylomicrons. These chylomicrons will go through the Golgi apparatus, and the Golgi apparatus will package chylomicrons into little secretory vesicles where it gets exocytosed. So chylomicrons are exocytosed. And due to its large size, chylomicrons cannot go simply into the capillary or the bloodstream. Instead, lacteals will absorb chylomicrons, and it's from there that fat will go through the lymphatic system and then enter into the vena cava, where it can go into the circulatory system. And in this last slide, I wanted to give a brief summary of what I talked about about fat absorption. So primary fat absorption occurs in the duodenum of the small intestine. Another key takeaway is that bile salts and colipase aid lipases in breaking down lipids into its smaller parts. Fat droplets are emulsified and broken down into micelles. Then these fatty acids, the monoglycerides, will travel through simple diffusion. Cholesterol will travel through a transport protein into the interiocytes. Chylomicrons are formed in the interiocyte, and then it gets packaged into secretory vesicles. The fat will then travel through the lymphatic system via the lacteals. And then the body will use fat or store them as adipose tissue. And that is the enzymology and the anatomy of fat absorption. All right, thanks.